So what we're going to deal with in this lecture is forward modeling and inverse modeling. That's basically also what we're going to do within the hands-on session that immediately follows this lecture. So the overview of the lecture is that I'll start with a short motivation and background. Then there's a section on forward modeling and there's a section on inverse modeling. But let's start with the motivation and the background. So the strong points of EEG and MEG, of course, are its high temporal resolution. Like we can sample as fast as we want, and we can really track brain activity at a millisecond scale. And that allows us to utilize this temporal information to characterize individual components of the event-related potential, or difference components such as the mismatch negativity that we've been looking at yesterday. And it also allows us to look at oscillatory activity. Such as we've been doing this morning. And of course, finally, what we'll be looking at tomorrow is also how you can use network analysis methods, connectivity analysis methods to disentangle dynamics of the networks, to look at the interaction between different brain regions. But as you by now are aware, the weak point of EEG and MEG are, of course, that it's a measurement on the outside of the brain, and that we have overlap of components. We have quite a blurry representation of cortical activity, and we have a very low spatial resolution. MEG, of course, has a much better spatial resolution than EEG, but still MEG suffers from a relatively low spatial resolution given the level of detail that we know actually about the brain and how different brain modules, different brain nodes are involved in certain tasks. So if you find an event-related potential or event-related field component, you want to characterize it in its physiological terms. And the time or the frequency are the natural characteristics of the data that we have. So we can use a lot of different methods, but it's always very straightforward to think about oscillations in the data because you can actually see them happening in the data as you're acquiring the data. But the location is much more difficult. Interpreting the location requires an interpretation of the scalp topography. And forward inverse modeling techniques are techniques that help you in interpreting scalp topographies. Furthermore, forward inverse modeling techniques helps to disentangle the overlapping source time series. So yesterday I showed this figure already. So where we have multiple sources, where the sources have different time series. And if we look at the scalp level, what we see is an overlap of the temporal profile of the activity. Of course, at some channels it's going to be less severe, but this is like an idealized scenario. You've already seen that there's a lot of overlap. So one of the key features of source modeling is that we want to locate the regions where the activity is coming from. But perhaps even more important is that we want to disentangle the time series such that we can say something about the time series. And that was also that point was also made by Caroline Witten yesterday in her lecture, where she showed that at the source level you can distinguish the cortical activity much better than at the channel level.
By the way, you now have your slides in your email.
Ah, thank you.
Thank you.
Okay, so the source modeling, forward and inverse modeling, has a few components in it. On the one hand, we have the physiological sources. So we have sources in the brain that are causing electrical currents, neuronal tissues, basically causing electrical currents. Then we have the volume conductor. So we have the tissue of the body, the tissue of the brain, the skull and the scalp, through which these volume currents will flow. And then we can make an observation of the consequence of these currents by attaching electrodes to the scalp or by measuring magnetic field. If we go from the physiological sources towards a model of the observed potential field, that's what we call forward modeling. And if we go from the observed potential or field to the physiological sources, that's what we call inverse modeling. Inverse modeling, that's where most of you will be interested in. Like forward modeling is more the field of physicists, inverse modeling is more the field of experimental neuroscientists. But we cannot do inverse modeling without forward modeling. That's also why it's important that you also have some understanding of how forward modeling works. So that's what I will focus on first. In forward modeling, we have to consider a few aspects. And the first aspect is the source model. This is something that you have already seen, although in a different slide. We have cortical tissue, we have neurons, and it's especially the pyramidal neurons which are nicely aligned if the synapses arrive from the pyramidal neurons, they produce postsynaptic potentials, and these postsynaptic potentials, they sum up to something that we can record. In forward modeling, rather than making a model for each of the individual pyramidal neurons, what we do is we summarize all these pyramidal neurons by a simplified model, which is the equivalent current dipole. The equivalent current dipole is a model that produces current, which is equivalent to the current that would have been produced by very detailed modeling of all these individual neurons. So if we're saying equivalent current dipoles, or if we leave out the equivalent current part, so if we say dipoles, it's a model for the brain activity. It's not that the brain has dipoles in it, but equivalent current dipoles are just a very convenient way of describing this in mathematical terms. So we have equivalent current dipoles with which we can describe activity in a small patch of cortex. But if we zoom out a little bit, so if you take a little bit a larger patch, then each of the cortical columns, for example, can be modeled with an equivalent current dipole. But again, if I take all of these small equivalent current dipoles, and if I look at the current and the potential distribution that they're generating, I can still simplify the model further by taking a single larger equivalent current dipole. So an equivalent current dipole can still model the activity of an extended patch. This idea starts to break a little bit if the patch gets too large, or if the patch gets very curved. Because what then will happen is that the equivalent current dipole is going to represent the center of mass of the activity in the patch, which is not necessarily in the patch anymore. So what you see here is that the dipole is offset relative to the patch. So that's just something that you have to keep in mind, is that the equivalent current dipoles that we're often considering in explaining brain activity, they model the cortical surface, but they're not always located on the cortical surface. And that's not necessarily a problem as long as you understand why they might be displaced. Besides the source model, we also need a volume conduction model. And we have a number of choices that we can make for the volume conduction model. The volume conductor describes the electrical properties of the tissue, and it describes the geometry of the tissue. So it describes it makes a geometrical approximation of the head. But it's important that the volume conduction model is really about the passive tissue. Like, it's really about the tissue that's just passing current through it, because the tissue is electrically conductive. So it describes how the currents flow, not where they originate from. And actually, we're using very similar or identical volume conduction modeling techniques to study TMS and TACS and TDCS than the ones that we're using to study activity that's being generated in the brain. The simplest volume conduction model, of course, that we can consider, and that has been used a lot in the past, is the spherical volume conduction model. And the spherical volume conduction model is a first approximation, but it doesn't really fit very well. If you look at it from this side, it seems to fit reasonably well, but what you should realize is that at the bottom of the brain, it doesn't really describe the geometry. And if you were to see this from the front, what you also would see is that the sphere actually extends quite a bit at the edges. So a spherical volume conduction model is not very accurate. So we have different computational methods for volume conduction problem that allow for realistic geometry. So for the spherical volume conduction model, there's an analytic solution, so we can compute it up to an arbitrary precision, and it's very fast, it's very efficient, but it's just not very accurate. So if you look at methods that we have for computing the volume, for computing solutions to the volume conduction problem using a realistic geometry, the first is the boundary element method. The second is the finite element method, and the third is the finite difference method. Now I will shortly explain the difference between these three terms, and they're called BEM, FEM, and FDM. That's how you would normally find them in literature. One of the differences is in the geometrical description that they take. So the boundary element method describes the boundaries between the important tissue types, and those boundaries are described with triangles. The finite element method is usually done describing the geometry using tetraheders. So the whole volume is tessellated, not only the surfaces, but the whole volume is tessellated with tetraheders. The finite difference method is usually done using hexaheders or using small cubes. Although nowadays also more and more research is being done using hexaheders in the finite element method. But it's basically the finite element and the finite difference method both use a volumetric description of the geometry, whereas the boundary element method only describes the boundaries. If we look at the boundary element method, it describes the geometry by the compartments, and each of the compartments has to be homogeneous, which means that the conductivity has to be the same all over the compartment, and each of the compartments has to have an isotropic conductivity, which means that the conductivity is identical regardless of the direction in which the current is flowing. So these are requirements of the boundary element method, and we know that these constraints are not really the case in the brane, but it's an approximation that we can make. So that means that if we look at the brane compartment, if we look at white matter, we know that it's not isotropic, because the white matter fiber bundles have a certain direction, and current flows slightly easier along the fibers than across the fibers. But this is an approximation. And the important tissues that we incorporate in the boundary element model are the skin, which basically includes not only the skin but also the subcutaneous fat and the muscle, the skull, so the bone, and the brain. And if we put a lot of effort in making the model, we can also include the cerebrospinal fluid. What we do is we describe the surfaces between the boundaries, the surfaces of the boundaries between these tissue types using triangles. And the reason for using triangles is that triangles allow us to use arbitrary complex geometry. So this is a triangulated cube, but I can also use triangles to describe a sphere, or I can use triangles to describe something that looks like a head. So triangles are just a very convenient way of describing a closed surface, because that's a requirement of the boundary element method. Like we have a compartment and we describe the surface around the compartment. The approach that we use in the boundary element method is that we start by constructing the geometries, usually by taking the anatomical MRI and segmenting in different tissue types. So if this is the anatomical MRI, then we use image processing techniques to construct something like this, and then using these using the segmentation, each of the voxels is assigned to one of the different classes of tissue, and then from the segmentation we can determine the surfaces that describe the boundaries. So the first step, and that's also what we're going to do in the hands-on session, is really more on the level of image processing, like working with the voxels, which first have ray value data describing the anatomy, and then converting it into a binary representation. Is this voxel skin, yes or no? Is this voxel skull, yes or no? And then once we have a binary representation, we can use techniques to make a mesh that describes the outside of the binary data. So the BEM, yeah? Oh, sorry.
Just briefly, if we don't have the MRI, what can we do with something more general?
Yeah, so the answer to this depends on whether you're doing MEG or EEG recording. With EEG recording, it's actually much more common not to have an MRI, because EEG systems are usually installed in places where you just don't happen to have an MRI scanner. MEG happens to be installed in places where an MRI scanner is usually like around the corner or so. So with MEG it's just easier to get an MRI done. With MEG it's also more important to get the MRI done, because with EEG what you're doing is you have electrode caps in different sizes. So if you have a small subject, you're just going to put on a small electrode cap, large subject, large electrode cap. So basically the placement of the sensor scales along with the size of the head.
So it's not a problem in EEG?
So what we do quite often in EEG is, if we don't have an anatomical MRI, is that we'll just take a standard anatomical MRI and we're just going to put electrodes on that anatomical MRI as if they, according to how they should have been, had this subject had this electrode cap.
So like the MNI template?
For example, the MNI template. With EEG, it's more important that you have a correct determination of where the electrodes are, because that's the difficult part. So having polyimage recordings of electrodes is actually quite valuable. It's perhaps more valuable than having an anatomical MRI. With MEG, however, it's different, because with MEG the sensors are fixed, and it's really the size of the head that changes inside the helmet. So if you don't compensate for the size of the head in your forward and inverse models, you're just going to get wrong estimates. So with MEG you cannot use a template, or well, you can use a template, but you really have to scale the template to the individual size of the individual subject. So that means that with MEG, at least you have to have a measurement of the size of the head. And if you can use a measuring tape, just measure the circumference. You can use a Polymus electromagnetic tracker to record the head shape. At least those will give you some numbers such that you can take a template, such as the MNI brain, and you could scale the template such that it matches the approximate geometry of your individual subject. Okay, so with the BEM, what we do is we segment the tissue in different tissue classes. We extract the surfaces, and what we subsequently do is we downsample the surface to a reasonable number of triangles. Like we can use a lot of triangles in the BEM, but it's going to make the computation very slow. What we need is we need to have the triangles, because the potential changes over the triangles, and the potential changes most rapidly on the interface between the brain and the skull. So what we do with the BEM is that we take actually the most triangles on the inside and the fewest triangles on the outside surface. To have most of the triangles there where it really matters. So with the BEM, it's not so important that the face looks nice. It's important that the inside of the skull looks nice. Subsequently, we compute the boundary element model, and that's a single computation. That computation is independent of the source model. It's one lengthy computation, and depending on the speed of your computer, it actually doesn't take that much time. But once you have the BEM model, you can use it for arbitrary source configurations. And that's really convenient, because it means that you only have to compute it once, and then you can use it for all sorts of experimental data that was recorded from this single subject. Yep. So the finite element method is another method. Rather than starting with the boundaries, it starts with tessellating the three-dimensional volume in tetraeders. Again, tetraeders are very convenient because you can describe arbitrary complex geometries with it. But the consequence is that we have a large number of elements to cover the whole brain and to cover the whole head. Each tetraeder can have its own conductivity, and each tetraeder can actually also have a conductivity which is anisotropic. So that means that with a FEM, you can model the anisotropic distribution of conductivity much better than with a BEM. What we subsequently often do is that we take more elements where the potential changes rapidly and fewer elements where the potential doesn't change that rapidly. So we're refining it where needed, and we're making it coarse where it's possible. The finite element method is the most accurate numerical method, but constructing these tessellated volumes is quite difficult. Like, we don't have so many useful tools for that yet. And it's also computation is much more expensive. So that means that the finite element method is, as of yet, is not being used that much. We are currently working together with the Symbio team in Münster in Germany on making the finite element method available in FieldTube. So it is already available, but it's still computationally a bit clumsy. So there's still a little bit more work to be done to make the finite element method really applicable to large-scale data sets. And then we have the finite difference method. And actually, the finite difference method is easy to compute, but it's not really useful. But still, I want to use it to explain to you how volume conduction modeling works. What I'm going to do is I'm going to show this template brain. And I'm going to split the brain, or I'm going to split the whole volume in voxels. And let's now zoom in on this voxel and on its neighbors. So if I consider this voxel, and this voxel is connected to its neighbors, and the connection is basically done with a certain resistance. So what I can think about is with a simple resistor network. I have a node that's connected to its neighboring nodes. If we have current that is flowing out of the node, and I also know that the current has to flow in from the other nodes, assuming that there's no current generated within the node itself. So that means that we have this simple equation. So the current flowing in or flowing out on one side is the same as the current as the sum of the currents flowing in at the other sides. And this is called Kirchhoff's law. It's just the law of preservation of current. Then we also have Ohm's law, which I guess most of you should know. And we can combine these to give me this equation. And what it basically says is that the current over resistor 1, so the potential difference over resistor 1 divided by its resistance, plus the current over resistor 2, etc., sums up to 0. So this gives me one equation. What I now have, I can expand that equation by taking all the potentials inside. And what it basically gives me is an unknown potential at each of the nodes, and I have a linear equation at each of the nodes. So let's say that we have about 100 by 100 by 100 voxels, which is a relatively coarse description of the geometry. And I have 1 million unknown potentials, and I have 1 million linear equations. Well, that's a linear system of equations that I'm able to solve, because I have as many unknowns as I have equations. What I subsequently have to do is I have to add a source and a sink, because this set of equations only describes how current flows. It doesn't describe where the currents come from. So what I do is I say, well, the sum of the current is 0 for all the nodes except for one node where current is being injected, and another node where the current is being drawn towards. And of course the question is like, how does the current from this node to this node, how does it flow? So this is basically a set of linear equation, and I can solve for the unknown potential. The final difference, so like what I'm saying is I have 1 million unknown potentials. So in principle, it's a very large linear system, but of course it's a very sparse linear system as well, because each node is only connected to its neighbors. So we can use sparse matrix algebra to solve this relatively efficiently, but still it's a clumsy method. So that's why it has not received widespread attention. The methods that we have implemented in FieldSub are available. Sorry.
When you say it's a clumsy method, what do you mean? What's wrong with that?
It's very memory intensive. So even if we use a sparse representation, it requires to represent a lot of linear equations in memory, and then the matrix with all these equations has to be inverted. So what we're doing is we're doing an inversion of a very large matrix. The size of the matrix depends on the description of the geometry. So if we take 100 by 100 by 100, then we have voxels of approximately 2.5 millimeter large, which means that if we try to model the skull, then we only have, especially over temporal cortex, we only have one voxel, which is a very coarse representation of skull. So we would need many more voxels to get the geometry described properly. And it's very difficult to get the geometry described correctly if you use voxels. That's why the finite element method uses tetraeders, and the boundary element method uses triangles, because they're much more flexible in describing the fine, detailed anatomical structure.
And there are different sizes of tetraeders.
Yeah, you can make small ones and large ones, and you can just scale them locally, and they still fit together. Whereas with voxels, it's difficult to make voxels in one region of the brain smaller, to make them larger in another region. It just wouldn't fit. Furthermore, another aspect which is clumsy is that in order to solve the finite difference method, I have to take the sources into account. Whereas both with the finite element method and the boundary element method, I'm solving the volume conduction model only once, irrespective of the sources. And here, I'm solving the volume conduction model given the sources.
So each time?
Yes. Although there are some tricks to make this easier, but it's not so easy to keep the separation between the source model and the volume conduction model in the finite difference method. So that's what I mean with clumsy. Okay, so let's have a look at the methods that are implemented in FieldSub, and this is also what we're going to practice. So what we usually would start with is by reading the anatomical MRI. Then we would call the FD volume segment function after some processing of the anatomical MRI. And then we would call FD prepare mesh. And then, depending on the method that we would use, would tessellate the brain, the skull, and the scalp, and we would use different numbers of vertices for each of the surfaces. And then we can call FD prepare head model. And an FD prepare head model will make the actual head model, will actually make the volume conduction model. And one of the methods that you can specify is concentric spheres. Oh, sorry. what concentric spheres will do is it will just fit a sphere to each of these meshes. So what this is going to do, it's going to fit 3 spheres that have the same center, and then you can use that as a volume conduction model. You can also use the boundary element method, and we have a number of different implementations of the boundary element method. The BEMCP, that's an implementation by Christophe Philippe. It's a pure MATLAB implement, although it uses MEX files, but it's a MATLAB implementation, and it's a relatively fast method, so that's why it's quite popular. But it's not so numerically accurate. We also have an implementation which is called DPoly. It's an implementation by a colleague of mine in Nijmegen, by Tom Oostendorp. The problem with the DPoly implementation is that it's only available on Linux computers. It's because it's an external executable. We have a third method which is called OpenMEG. Now, OpenMEG is a large project from a French research group. Also, OpenMEG, although being available on Windows, Linux and Mac computers, requires an external binary, and it requires that you have to go through a more complicated installation procedure. So if you're serious about doing volume conduction modeling in EEG, then I recommend that you use the OpenMEG method, because it's the most numerically accurate, and it will for sure run on your computer. But it just requires some investment to set up the software on your computer.
So you can run it on FieldTrip then?
You can run it from FieldTrip, but FieldTrip will then write temporary files to disk, and FieldTrip will call the executable. So in the background, it's going to run the executable. So it's not something that you have to interact with the software, but you have to make sure that the software is installed at the proper location, and you have to set the path properly.
I bet you do yourself.
And that's what you have to do yourself. So it's not included by default in FieldTrip. So you have to download it separately and then install it separately. For the finite element method, we have the Symbio method implemented, and that allows you to make really accurate volume conduction models, but at this moment it's probably still too slow to be practically usable, but that's something that we're still working on. I want to have a little bit of time to compare EEG and MEG, because they do require different volume conduction models. And this is a slide that Stephen already showed on Monday. So what we have is we have the brain, and we have activity in the brain, and that activity is causing currents to flow through the skull, through the CSF, the skull and the scalp. They reach the electrodes which are attached to the scalp, where we record them as potential differences. What we also have is we have the current flowing and the current producing magnetic field, and we can pick up this magnetic field with an MEG sensor. However, this is not the full story, because what we have is that the MEG, so for EEG this is the full story, but for MEG it's not the full story. With MEG what we have is we have currents, and currents produce magnetic fields. But we don't only have the current of the primary source that is producing magnetic fields, but also all of these currents are producing magnetic fields. So that is why with MEG we still have to consider volume conduction model, because it's still the currents that are flowing through the volume that are producing magnetic fields. And some of these currents that are flowing through the volume actually are very nasty. If you consider the primary current running in that direction, it's right-hand rule, I would get the field rotating like this. But if I look at how the return currents are, then basically they loop like this, which means that over here, current is going in that direction, which means the field is going like this. Which means that I have one field going like this, and the other field is going like this. That means that I have two fields with opposing magnetic field. I have two contributions to the magnetic field that are opposing each other. So with MEG, the magnetic field of the primary current is partially cancelled out by the sum of the magnetic fields of the volume currents. And there are certain configurations of the sources where it's not a partial cancellation, where it's a full cancellation. So if the source is in the center of the brain, and if the brain is a sphere, then we have full cancellation. In that case, the magnetic field of the primary current is just as large, but opposite to the magnetic fields of the volume currents, which means that there's no magnetic field outside the volume. So regardless of the sensor design, we're not going to record anything. And the same holds for a radial source. So if we have a source that is pointing towards the surface of a spherical volume conduction model, then the the secondary currents produce a magnetic field that's exactly opposite to the magnetic field of the primary current. Well, luckily, the head is not a sphere, so that means that we never really have full cancellation, but we still have that sources that are oriented towards the scalp towards the skull surface have a much weaker visibility outside than sources that are tangential to the skull surface. And also sources that are deep have a much weaker visibility than sources that are superficial. And the more effort we put in the volume conduction model, the better we will be able to see the radial source and to see the deep sources. So that's why it's important also with MEG to make accurate volume conduction models. So if you don't, then we're not going to see a lot of the activity. Yeah? Oh yeah, that's actually the. Oh yeah, this is slightly different. So this is one aspect. So the visibility of the sources, like just making sure that you understand that we also have volume conduction here. The second is that with EEG, what we're recording is potential difference at the scalp, and these correspond to the current flowing through the skin, but only a very small fraction of the current passes through the skull, and therefore we have to model the skull and the skin as accurately as possible, because they have a lot of influence on how the potential distribution is going to look like. With MEG, we basically have the same situation, so only a tiny fraction of the current passes through the polyconductive skull, but therefore we usually ignore the skull and the skin in the MEG volume conduction model. The volume currents in the brain are still important, because it's those that determine whether a source is more or less radial or whether a source is relatively deep. So for MEG, we still have to describe the volume conduction properties of the brain compartment, but we don't really care too much about what's happening outside, because the currents are so small anyway, we can ignore them. And the consequence is that making a volume conduction model for MEG is much simpler than making an accurate volume conduction model for EEG, which is something that you'll experience in a hands-on session. At the same time, it's also important to stress the similarities between EEG and MEG. So we have an identical source model. In both cases, we're assuming that we have equivalent current dipoles. We have a very similar volume conduction model, and the identical inverse methods apply. So whatever I'm going to tell about in the second session about the inverse methods, it works both for EEG and for MEG. You have to keep in mind that for EEG, you're always recording potential difference. So that also means that you have to consider the referencing scheme. And the referencing scheme, the way that you have referenced your data, has to be consistent with the way that the forward model is referenced. And actually, convention is that with EEG volume conduction modeling, we always take a common average reference. And the motivation for that is that with a common average reference, we're averaging out part of the model errors. So with EEG volume conduction modeling, EEG inverse dipole fitting procedure, you always have to do a common average reference on your data. Yep.
Does reference electrode position play a role?
No, not so much anymore after you have re-referenced the data. If you do a recording with the reference electrode, then of course you know that by construct the potential at the reference electrode is 0. If you then subsequently re-reference the data, of course you can explicitly insert the reference electrode in your data set with zeros and then re-reference, because it's still, you have information about that electrode. So I would include the reference electrode in the EEG data set, although I realized that I didn't do it for the hands-on session. But in general, you can insert the reference electrode with all zeros. There's an option in FT pre-processing for that. And then you don't lose the reference electrode during the process of re-referencing. Any other questions about this? No.
As I understood, when you make the forward model, you get this matrix, which is a linear model. I'm wondering if it's a linear model, and I assume it's not frequency dependent, why sometimes people say that a high frequency activity gets attenuated faster than low frequency in the brain. Why is that not true?
That's not true. So it's the attenuation. Well, a high frequency activity is usually much more focal, because it's much less consistent over a large part of the brain.
It's a spatial thing.
So it's a spatial thing that it's much more focal, and that's why it's more difficult to pick up at a distance. The low frequency activity, there's basically these very large parts of the brain that oscillate in synchrony at these lower frequencies. So that's why they are much easier visible on the outside of the brain. But if you just consider the passive electrical properties of the brain tissue, they're basically up to like 1,000 hertz or so. There's no difference in conductivity. Okay, so what I want to move on is with inverse modeling. And what I'm going to do is I'm going to cover 3 methods for inverse modeling. But one is, and the idea of inverse modeling is that we start with the observation of the potential of the field and we try to infer which physiological sources were causing that observation. But before I start explaining inverse modeling methods, I want to do a quick demo. So, this is a fish. It's called an elephant fish. That's because it has a long snout. And the elephant fish lives in the river the Nile, and it's living in very muddy water. And the way that it's navigating in this water is that it's using something very similar to echo navigation. So it's emitting electrical pulses along the sideline, and it's sensing its environment by recording the electrical potential. So what we did, we got ourselves an elephant fish, and we put it in a fish tank. And what you see is that we attached a lot of electrodes to the fish tank. And every time that you hear one of these clicks, it's the electrode, it's the fish basically sending out a small pulse. Yeah? What you see is that we hooked it up to a relatively old-fashioned EEG system. So this is the recording that we did like more than 10 years ago. But at that time already the EEG system was really old-fashioned. But it was just perfectly fine for this purpose of the demonstration. So it's a 64-channel EEG system, and what we do is we record the potential at many different locations of the fish tank, and we made a volume conduction model of the fish tank. Using this volume conduction model, of course, you can start asking the question, so where is the fish? It's easy because you can look through the fish tank, but just using the same methods as we're using for source localization in the brain, and over here you can see the computer running. You also can see that it's like a 10-year-old computer, still with a CRT screen, etc. We were fitting dipoles to the potential distribution that we recorded over the fish tank. So this fish is very much has a field distribution which is very much similar to a dipole, so that's why we modeled the fish with an equivalent current dipole. And what you see here is the equivalent current dipole being plotted in relation to the fish tank. So over here is the heating element, just as a point of reference, such that you can distinguish left and right. And what you see is that we're not only localizing the position of the fish, but we're also determining the orientation of the fish, like in which direction it's swimming. And what at that time was actually quite cool. It was like real-time source localization, where the position and the strength was constantly being estimated. So what I'll now show is like that we're not only doing this, but also that it matches reasonably well with where the fish is. So let's wait a little bit until it zooms out. Okay.
So if you would model the fish, it would be 3 triangles.
It's just it's a point. We're modeling the fish as a point. Okay, so let's now have a look at where the fish is in the fish tank, and let's try to compare it with what you're seeing here. So this is the front view that's most easily to compare. So what you see is that the fish is in the lower left corner. It's pointing towards the back. It's rotating. It's starting to point towards the right, and it starts moving alongside the back of the fish tank. So it's working quite well. So this is basically what you have to remember if you're doing source modeling. It's really all about like a simple question: given a spatial topography, where's the fish? But then in the brain, we don't have a single fish. Yeah. In the brain, we have a lot of neurons. And actually, so we did this for a science project where we invited like a large audience to show people how electricity is being studied in all sorts of different domains. And for the project, we also tried to do it with 2 fish at the same time in a fish tank, and that didn't work. Because in the brain, what we have in reality, we have a lot of different neuronal activity. So that means that, and we cannot look in. So that means that locating cortical activity in the brain is much more challenging than locating a fish. But basically, the technique is very similar. So even if you forget all about all the mathematics that I'm going to show next, just remember this demo, okay?
You know how the fish navigates with using its electrical.
I don't know. We did not check with the biologist on how actually it's just a feature that we used of the fish. I don't know much about the fish
If we
itself.
had 128 fishes, maybe we could kind of have an aquarium and have fishes.
Yeah, the fish will actually point to the location of the cortical activity. Because the fish know how to interpret the environment. Right. Okay. Ik heb toch nog steeds 15 minuten?
Nee, je bent nu is hij klaar.
Nee, ik heb het is 2:15. Ja. Steven is trying to hurry me through the lecture, although I still have.
Ja, het is mij het begint om 1:30. Nee, 15 minuten, je hebt gelijk, sorry.
Okay.
Ik was hier eventjes niet.
Yeah.
Skip picture.
Okay, so what I want to do next is I want to walk through the different methods that we have for inverse modeling. And these inverse modeling techniques, they can be split over 3 different categories depending on the types of assumptions that we're making about the sources. And the first method that was developed in the like halfway the 80s is dipole models, like dipole fitting procedures. And the idea is that we minimize the error between a model and a measured potential. The second category of models that was introduced at the end of the 80s, like early 90s, is distributed dipole models. And with distributed dipole models, we have a perfect fit of the of the model to the measured potential, which means that we need additional constraints on the source. Now I'm now quickly going over it, but I'll give more detail in a few slides from now. So we have a number of different additional constraints that we can have, such as smoothness or power or amplitude. And the third family of source reconstruction methods is our spatial filtering methods. And with spatial filtering methods, we compute the filter output at every location, given an assumption on the time courses of the different sources. And different methods for spatial filtering are, for example, beamforming, such as LCMV, linear constraint minimum variance beamforming, or DICS, or multiple signal classification. So if you just conceptually think about how these methods work, what I have on this side of the figure, I have a true source. The true source projects onto a number of electrodes, and on this side of the figure is basically my model space. So what I'm doing with dipole fitting is that in my model space I'm moving a dipole around until the computation that I do on the electrodes for my model matches the actually observed potential of fields. With distributed dipole models, what I assume, well, there's potential activity everywhere, at least everywhere in the gray matter. So I have a lot of dipoles. And then given all these dipoles, I can compute the contribution of each of the dipoles to the magnetic field, and I can estimate how strong each of the dipoles would be. But since I have many more dipoles in my model than I have electrodes, I'm always going to get a perfect fit. And with spatial filtering, I'm estimating activity at one location at a time, and I'm moving that location through the whole brain. And every time again, I'm computing the spatial filter again and again. The methods that we have implemented, so all of these methods are implemented in FieldTrip, and the way that these are implemented is by the FT dipole fitting function. FT source analysis, where you specify config method is minimum norm estimation, so this is for distributed dipole fitting, or where you specify config method is DICS, dynamic imaging of coherent sources, which is a frequency domain beamforming technique, or where you specify linear constraint minimum variance, which is a time domain beamforming technique. So these methods, they all share the FT source analysis function because they all start with a distributed representation of the activity. So they all start with a cortical sheet or with a 3D grid, whereas this one doesn't start with a distributed representation of the activity, but only point-like. So that means that also the output here is different from the output that you will get here. So the procedure that we use for source reconstruction in general is we start with the design of an experiment, we measure the brain activity, we do the data processing, pre-processing, etc. And that's basically done using these functions. We do time frequency analysis or time locked analysis. That's what we do with these functions. This is what you have seen so far. This is what we've been working on yesterday, and that's what we've been working on this morning. What we subsequently do is that we have to prepare the necessary geometrical objects. We compute the forward model, and that is done using FT-prepare head model, FT-prepare source model, FT-prepare lead field. And then in the source analysis, these two come together, and we take the functional data and anatomical data, and we do FT source analysis or FT dipole fitting. So this is again like thinking about the whole problem as like in terms of a flow chart. How do these functions from the field toolbox, how do they fit together to make an analysis pipeline? Let's go over the different inverse methods one by one. So single and multiple dipole fitting. The idea of single and multiple dipole fitting is that we manipulate the source parameters to minimize the error between the measured and the model data. And the source parameters that we can manipulate are the location of each of the sources, the orientation and the strength. And the orientation and the strength can be expressed as a vector, and we call this vector the dipole moment. And it means that we can estimate them linearly, we can estimate them jointly, and I'll explain that in a few slides. However, the relation between the position and the scallop distribution of the potential is non-linear, so that also means that the position has to be estimated non-linearly. But it's important to realize that dipole fitting is source parameter estimation, and you're all familiar with parameter estimation. So this is just a scatter plot of measures of y that were observed at certain locations x. What we can do is we can make a model, and this model describes how the data y is explained depending on data x given certain model parameters. And that's basically what we do with dipole fitting. So we have the parameters. We have a source model with few parameters, the position, orientation, strength. We compute the model data and we minimize the difference between the actual and the model data. So what we do is we try to determine the parameters of the dipole model given the data. So in this case what we would do is we would estimate the slope and the intercept. The ingredients that we have are the forward model, which consists of the volume conductor and the source, and we have the measured potential at each electrode. So let's indicate the measured potential by y measured, and we have a model for the data, y model. Because the potential depends on the location of the electrode, x, and the model potential depends not only on the location of the electrode but also on model parameters a, b, c, etc. So what we then do is we determine the optimal model given the data. So what we're going to do is for each electrode we're computing the difference between the measured potential and the model potential. We take the square root of it and then we sum it over all the electrodes. So that's an indication for the error. It's just a sum of squares, and then we minimize the sum of squares given the model parameters. Yeah?
It's always a linear additive model?
Yes. And yes, that's what I'll deal with now. So what we have is we have linear superposition. That's just because the volume conduction properties of the brain are linear in the frequency domain that we're considering. So it means that if we have multiple sources, they just add up their potential. So that's convenient. If we have three sources, then these three sources, the potential of these three sources combined is just the sum of the individual potentials. And that is convenient to think about this dipole moment. Because what we have is we have a position at which we have current, but this current can be decomposed in three orthogonal directions like x, y, and z. And then with the current in the direction x, y, and z, we can make a current vector, which is the dipole moment. So that means that the potential that we have for a dipole is the strength along the x direction times the potential distribution for a dipole along the x direction plus the strength along the y direction times the potential distribution, etc. So this is something that I can write in matrix notation, where this is a matrix that we often call the lead field matrix, which is the number of electrodes times 3, which we then multiply with the dipole moment. So the model potential is this matrix times the dipole moment. So let's call this lead field matrix L and the dipole moment Q, what we have is we have a model for the data, which is L times Q, where L depends on certain source parameters, and L depends on the position. So I'll just keep the position implicit here in this zeta parameter. But it also means that we can invert this model. So what we can do is we can compute the inverse, or we can compute the pseudo-inverse actually, of this matrix, and we can multiply the potential with it to give us the estimate for Q. So that means that for a fixed position, the estimation of the dipole moment is a linear procedure. And that's nice because linear procedures are robust and they're fast. It means that we still have to deal with the nonlinear parameters in the model, and that's the position. So if we again look at the error term, what we have is we have the sum of squares. What we want to have is we want to have the minimum of the sum of squares given the position parameter. So again, position parameters are like positions along the x, y, and z direction. But now we have to consider how this error landscape looks like depending on the position. And it's difficult to visualize because it's a 3-dimensional or a 6-dimensional error landscape. But what you can imagine is a landscape where error can go up and can go down depending on how well a dipole at that position is able to explain the data. Of course, at every location, assuming that you have the optimal dipole moment. And what we have to do is we have to find the location of the parameters where the overall error is the smallest. And for that, we're using gradient descent methods. So, if we would have one dimension over which we had to estimate a parameter, for example, the location along the medial lateral axis, we could, for example, take 100 possible locations, and for each of those locations we could compute what the error is, and we could take the minimum error. If we have two locations, sorry, if we have two dimensions, so medial lateral and inferior superior, then we would have 100 times 100 locations over which we would have to compute the error. If we have 3 dimensions, then we would have 100 times 100 times 100, which is 1 million locations over which we would have to compute the error. Okay, so each of these computations takes time. Let's say that it takes 1 millisecond. It means that if we want to scan the whole brain at a resolution of about 1 centimeter and compute the error everywhere, it's going to take about 1,000 seconds. Well, 1,000 seconds is actually not too bad. It's close to 20 minutes, so that's doable. But what if we have 2 dipoles? Suddenly we're looking at not 1 million possible locations where we have to perform the computation, but we're looking at this number. So that means with 2 dipoles, I cannot scan the brain with 1 dipole and then for every location also scan the brain with the other dipole. It would take 1,000 times 1,000 seconds. Just take 1,000,000 seconds. That's too long. I'm not going to wait for that. So that means that if we have dipole models in which we have more than a single dipole, we have to use other strategies than simply scanning the whole grid. And those strategies are basically these gradient descent methods. So what we do with the gradient descent method is that we start at a good starting point and we allow the dipole parameters to be changed such that the error gets smaller. However, there is no guarantee that the dipole is always going to end up at the location where the error is minimum. So it might very well be that the dipole is going to end up here, which is a local minimum. So in the nonlinear optimization procedure, you always have to be aware that the location that you're getting might be a location where the error is locally minimal, but not the global minimum. And of course, this is the global minimum that we're after. But the problem is we cannot see the error landscape. So that's why we often use different strategies. We use different starting points. And if all the different starting points end up at the same final location, then we can be a little bit more confident that it's not a local minimum. Yep.
I was thinking for patients with epilepsy, we have a spike, and biologically we would assume it's from one place.
Mm-hmm.
But would it be safer to do like a grid search?
Oh yes, yes. In those cases, I would certainly consider a grid search to be sure that you don't end up in a local minimum. Yes. Yeah. Yep. Actually, what we often do is that we start with a grid search, and that we subsequently refine the location, starting from the grid position which is the best, because then you can start with a grid search on a 1 centimeter grid, and then you can subsequently refine the position even more. Okay, so the data that we're working with is data that has different types of channels. And already yesterday I had the question like, can we combine the different types of channels in these types of analysis? So what we have to consider is that we have some channels that are electrodes. And the data that we have on these channels is in microvolts. We have some channels that are magnetometers, and we have some channels that are gradiometers. So that means that if we compute the sum of the squared differences, we're adding squared apples and squared pears. It just does not add up. So what's going to happen is, if we would do this, that the channels that just happen to have the large numbers are going to influence the results most. And the channels that happen to have small numbers are not really going to influence the results. So combining channels, combining different channels with different units on the channels is not trivial. Of course, what you can do is you can apply a weighting factor for each of the different types of channels, and then you can combine the different channels in one source analysis strategy. But that's not something that we're going to work on today.
Could I ask?
Yep.
Is it okay?
Yep. Yeah, it depends a little bit on Stephen. Stephen is a bit tight on time.
Yeah. Well, no, let's do it during the
Okay,
hands-on.
yeah.
Yep.
Yeah.
Okay. Okay, let's move on with distributed source models, because the assumption that we're making with single and multiple dipole fitting is that we have only a very few points in the brain where the activity is coming from, and actually we already know where the activity is coming from. We know that the activity is coming from gray matter, so why don't we just model gray matter? So the assumption in a distributed source model is that we already know where the activity is, but we only have to estimate the strength of the activity. So what we start with is a predefined grid, which is either a three-dimensional volume, or preferably it's a cortical sheet. It's a description of the gray matter. And at each of the elements of this grid or of the cortical sheet, we estimate the strength. So that's in principle, it's easy to solve because it's just a linear problem. Right? Like I already explained how to do it with 3 dipoles, in estimating dipole moment. So I could also do this with 1,000 dipoles. However, we have many more unknowns, parameters that have to be estimated, and then we have knowns, like data points. And that means that we have an infinite number of solutions that is able to explain the data perfectly well. And that's why we need additional constraints on these solutions to come up with a unique solution. But in the end, it's just a linear estimation problem. So what we do is rather than modeling the cortical activity with a few dipoles, what we do is we take a cortical patch, and we, at every triangle or at every vertex, and in this case it's every triangle, we place a dipole. Normally we would place the dipoles at every vertex. And then we try to estimate the strength of all these dipoles simultaneously. And this is the mathematics behind it. So it's basically the same equation I showed before, except that we now have many more sources that are contributing here, which means that this lead field matrix is the number of channels by 3 times the number of sources, which is a lot. So it's again, we use the same inversion procedure, but we have a distributed source model with many dipoles through our whole brain. If we estimate the strength of all the dipoles, data, but also the noise is going to be perfectly explained. What we have in this parameter estimation procedure, something like this. We just have a model that, regardless of what kind of noise we have, it's going to explain the noise perfectly well. Which means that if we don't do anything about it, that's just going to result in very undesirable characteristics of the source estimate, because we're just overfitting the data. So the solution to this is to do regularization, to use a regularized linear estimation procedure. So this is the model that we in general assume for in forward modeling. So we have the data, which is the lead field times the sources plus some noise, plus some noise term. And it means that if we just take a very large lead field matrix and estimate Q, we won't get 0. But we can add an additional constraint. And that additional constraint is that not all of the data should be explained. So rather than saying, well, I'm only going to minimize the data terms, I'm also going to add a term that penalizes something about the sources. So what you see here is I have the sum of squared errors between the channels, measurements on the channels and the model on the channels. But I also have a term with which I can penalize the source solution. So what I can do is here I can implement additional options, and then using this lambda parameter, I can determine how much do I want to weigh the solution towards explaining all the data, or how much do I want to weigh the solution towards conforming to my a priori assumptions. And these a priori assumptions are, for example, implemented in this matrix D, where I can implement a Laplacian, like a smoothness constraint, where I can just implement identity, where I just get a minimum norm constraint. So basically we balance the trust that we have in the data with the trust that we have in the model. Yeah? And this is actually quite a difficult topic on itself, so it's something that we could easily spend a whole lecture on. But what I want to do is like move on with spatial filtering, because spatial filtering is a technique that's actually also very popular. It's used a lot in MEG, and one of the spatial filtering techniques that you already have heard a lot of is beamforming. Again, with beamforming, the position of the sources is not estimated as such, but we manipulate the filter properties, so not the source properties. So with dipole fitting, we make explicit assumptions on the sources. With beamforming, we don't make explicit assumptions on the sources. However, we do make an implicit assumption that the source can be modeled with a single dipole. In beamforming, we rely on additional assumptions about the data. And that is the most important assumption, is that multiple sources that are present in the data should not be too strongly correlated. The concept of spatial filter is basically that we're trying to zoom in on a piece of the data. With dipole fitting, we can estimate the dipole moment. And with dipole, if we estimate the dipole moment, as Caroline showed yesterday, like showing the time course of the dipole fitting, you can see this as a very poor spatial filter. Like it's a poor man's spatial filter. It works, but it's not very efficient. And what we do with beamforming is we just make much better use of the zooming capabilities of the mathematics that we have available. And that's something that I will explain in much more detail tomorrow. But the idea is that we have a forward model with a source that projects onto the channels, and we want to know, like, how do we combine these different channels to get an estimate of the source strength, given that the sources are not too correlated. And this is basically the spatial filter weights. So how do we determine the spatial filter weights? And tomorrow I'll explain how to get these spatial filter weights. So to summarize, what I've dealt with is forward modeling, which is required for the interpretation of scalp topography. And as soon as you start interpreting scalp topography, then in principle you are doing source estimation. And these mathematical techniques that I've been describing, they're there to help you in interpreting these scalp topographies. And that's basically that's inverse modeling. In inverse modeling, we have to make assumptions on the volume conduction model about the geometry and about the conductivity. And we have to make model assumptions about the source. Usually we say, well, we take a dipole, and a dipole is usually seen as a building block, so we can take one dipole or two dipoles, we can take 1,000 dipoles. So that means that we make an additional assumption on the source, either that we have a single one, or that we have multiple but not too many, that we have a distributed source model, or that we have, and that gives rise to different mathematical approaches. So dipole fitting would be used if we have a single point-like source or multiple point-like sources, and linear estimation would be used if we assume that we have a distributed source model. Spatial filtering can be used in both, but makes the assumption on the data not being too strongly correlated. It's important to keep in mind that on the one hand we're trying to estimate the location of the sources, on the other hand we're trying to estimate the strength of the sources. And I think that I already made the point yesterday that location is important, but location is mainly important because it allows us to infer the cortical dynamics. And the whole motivation for recording EEG and MEG is that we're interested in the cortical dynamics. We're interested in these time course, we're interested in these oscillations. So the main reason for us to do inverse modeling is not necessarily to find these spots, although with epilepsy research, with epilepsy diagnosis, it is important to find these spots, because those are the spots that you would surgically target. But in non-invasive electrophysiological research, it's not so much about finding the exact spot, because for that we have fMRI, but it's much more, given the channel level data, we want to infer what the source level activity is. That's what we use the source analysis methods for. That's also something that we're going to practice in a hands-on session. Are there any remaining questions?